Left off at slide 6. Take it away.
I'm going to start this slide at the top here for
Sure.
Ichthyopterius multifilius. So sorry, we kind of cut out here.
One second. Let me just verify that we're live. I want to verify that we're still good. And okay, like Facebook is once again going. Okay. Like we're live on YouTube again. Sorry about that, everybody. We're back. We had a network glitch on our end, but we reset the stream and we're back to continue our protozoal parasite discussion. So, Dr. Nick, I'll let you take it up where I think we left off.
Okay, I'm going to start again with Ichthyopterius multifilius because this is the slide that it died on. So Ichthyopterius multifilius is different from the other ciliated protozoa because it has a much more complex life cycle. The name itself, ichthyo, means fish, therius means louse, multi means many, and filius means son. So it's a fish louse with many children. And in this particular picture of a koi with the ich, you can see all the classic white spots. Now, the white spots will get on the fins, on the skin. They can even get on the gills of the fish and can cause a lot of damage to the gill tissue. When you do a skin biopsy on this fish and look at the tissue under the microscope, this is what you're looking at, these individual cells. And you see they're different sizes because they start out as very small and they grow as they feed on the fish and they get bigger and bigger. Most ciliated protozoa, they're one size. They eat to the point where they're healthy. They'll split into two pieces. Those two pieces become individual organisms and they start the cycle all over. With ich, it's a different life cycle. These little white specks on the fish are actually the parasite embedded under the epithelium, so under the skin of the fish. They grow to be about 1 millimeter in diameter. So that's why you can see these white bumps, which is the parasite covered with skin on the fish. And these will drop off the fish after they fed to be a certain size. And we mentioned the macronucleus, which is this U or horseshoe shape white part, and then the micronucleus, which is this little white dot right here. So we mentioned that the ciliates have a micronucleus and a macronucleus. So after the trophont, which is the feeding stage on the fish, gets full, it drops off and sinks to the bottom of the aquarium. There it can get onto plants, it can get onto the rocks, it can get on the gravel, or even attached to the glass of the tank. So if you're buying plants that came in from an aquarium that had fish, those plants could potentially be carrying this tomont stage of this parasite on the plant. So that's something to be aware of. But the tomont stage gets a hard case around it. It'll then start dividing. So you can see a little line right here, splitting it in two. And here's this white spot here is half the macronucleus. Here's the other half of the macronucleus. And they'll split into two pieces. And then from here, it'll go to 2, 4, 8, 16, 32, 64, 128, etc., until it divides 10 times and ends up to be a 1,024 cells. So each one of these tiny little cells inside here is one of the divisions of this tomont, dividing into multiple smaller particles. These particles are called tomites, and the tomites will split through the shell and emerge. And so this picture is a tomite here. It's got a little pointed end. It's cilia all over it. But this is very, very small. So if you look at the two pictures, it looks like they're about the same size, but actually this is magnified. I don't know the exact dimensions, but this might be 1,000 power magnification, and this one might only be 100 power magnification. So you see they're significantly smaller at this tomite stage. The tomite swims around the aquarium looking for a fish. And depending on temperature, it usually only has a day or maybe a couple days before it finds a fish and attaches to the fish. And if it doesn't get on a fish by that time, it will die. So that's one good news. It's the only stage of the parasite that's also susceptible to the medications we use. So if you're treating your aquarium with medication, And you're only killing this stage. You're not killing the white spots on the fish, and you're not killing the encysted stage here either. So if you only treat it one time, you're only getting rid of this one phase, and the others will continue to reproduce. And that's why sometimes people have a hard time treating ick, because they haven't treated it long enough to get every portion of the cycle through this tomite stage. So the question is, how long do you have to treat it? So if we look at this little chart here, the trophont time to maturity. So the trophont is the feeding stage. So when the tomont attaches to the fish, it becomes a trophont. It starts growing until it gets to be big enough to drop off the fish. If you're in an outdoor koi pond and it's 50 to 60 degrees, it could take 2, maybe 4, 5 weeks for that to occur. So if you're only treating it one time, you'll never get rid of this. You may have to treat it every week for 4 or 5 or 6 weeks in order to totally eradicate it. The other option would be in like our aquarium, where we're keeping it about 70 to 80 degrees. 70 to 80 degrees in a freshwater aquarium, it's going to take 3 to 5 days to feed. So you have to treat this every at least 3 to 5 days in order to get the parasites as they drop off. But then they go into this encysted stage, which is the tomont. So now we look at how long does the tomont take. So again, if we look at a koi pond, maybe 40, 50 degrees, it's going to take 5, 6, 7 days for this stage to occur where it's reproducing. If we're looking in an aquarium, maybe 70 to 80 degrees, we're looking at 1 day, 6 hours to a day or so. So that's not so long. And then it's going to release these tomites. And once it releases the tomites, that's the period you can treat it. So my recommendation is in a koi pond, you probably in cooler weather need to treat it at least once a week, and you need to treat it for 4 to 6 weeks. In warmer weather, you probably treat it twice a week and treat it for about 3 to 4 weeks. For a home aquarium where it's indoors and warmer, I like to treat it every other day and treat it for about 2 weeks. So 2 weeks treatment, let's say, Monday, Wednesday, Friday, and then Monday, Wednesday, Friday, or Sunday, Tuesday, Thursday, something like that. But you want to treat it every other day for about 2 weeks in order to hit this tomont stage throughout that whole period. Now, one last point that has been used before is if you do have a fish in a quarantine tank, which is good, once you take that fish out, if you wait, depending on the temperature, so let's say freshwater indoor aquarium, 5, 6 days, any tomonts that have reproduced and released tomites, those tomites won't have a fish, and so they'll die. So you can eliminate the population by just removing the fish, but you have to keep the fish out for about a week or so in order to eliminate all the stages of the parasite. And in the past, that has been done in fish where you just move them from tank to tank. Every couple of days, move them to a new tank and leave the old tank vacant until all the parasites have died. If you got the room, that works, but the treatments work as well as long as you understand the timing. The rest of the ciliated protozoa, they reproduce by binary fission, which means one organism splits into 2 and they're done. And so you can treat those at any time and it should eliminate the parasite. Okay, so that's ciliated protozoa. Now we're going to go to Peritrichia. The most common one we typically seen is Epistolis. It used to be called Heteropolaria. So if you're looking in old literature, you might have to look for a couple different names for it. And these are vase-shaped protozoa with an oral opening, and they have just cilia around the mouth. And they're sessile, meaning they attach to a substrate, although they have an immature phase that might be free swimming, but then it attaches to a substrate, and the rest of its life that it will remain on that substrate. And some of these form branching colonies. Now, that substrate that they attach to can be a fish. It can be a frog. I've seen them on turtles. So any aquatic animal that can attach to that. Now, when it attaches to the fish, where it attaches to the fish will cause a lesion, which here it's called red sore disease. And that red sore is skin lesion, and it's probably getting infected by bacteria. The parasite doesn't actually feed on the fish. it feeds on debris that's in the water, but it will attach to the surface of the fish. And so while it's on the fish, it will breed. It'll separate into multiple, binary fission. So one becomes two. They can split into stalks, and that's why you'll see them attached to each other. And it's called an epibion, meaning that it's living on another animal, although it doesn't feed on the animal. So technically, it's not a parasite, but because it attaches to the surface of the fish or even the gills of the fish, it does cause damage, which then allows bacteria to get in there. And because they're filter feeders, eating things in the water, but they're very common in water with a high organic load, meaning a lot of waste, a lot of debris. So dead leaves in a pond or uneaten food on the bottom of the aquarium. So if you keep your aquarium clean, gravel vacuum in it, get all this debris out, you're less likely to have these parasites that require this organic debris that's in the tank. And as I mentioned, bacterial infections are common. So you can see this parasite. It's got this little bell shape or cone shape. Up at the top here, there's this little fuzzy area. Those are the cilia. And the cilia pull particles into its mouth where it feeds. And then they have these stalks that they attach onto the fish when they split. So this one has now split into two. You can see right here. So originally this was one that became two. Then each of those two split, these split. And so they become multiple organisms. This one is oh, wait, go back. Do my videos not work? That's actually a video, but it's not working. So on this particular one, the video showed the cilia here all around this. There's a little cilia, and the cilia beat, which causes the little debris in the water to swirl around here. Then it's kind of sucks them into the bell-shaped chamber. And so all these are little debris particles that it's picked up, and that's what it feeds on. So another one of these Peritrichus organisms is called Vorticella, and it's very similar. It has the bell shape, it has the cilia around the mouth. The difference is this one has a stalk that has a contractile myonemes, which allow it to contract the stalk. And again, it occurs in water with high organic load, and the bacteria can grow where it attaches. So this video looks like it's going to work. And so here what we're looking at, this is a whole colony of these little things, and each one of them has a little thread that they'll attach to the surface of the fish. And if you notice how they kind of jerk a little bit, that jerking is the stalk contracting. And if you only had one of these, it's really easy to see it, like on a microscope slide, when you do a scrape off the fish and you look at it, you'll see him just kind of pull back on the attachment site. So that's what that little motion was there. So the next group, we're talking about flagellated protozoa, flagella meaning whip. And so these flagellateds have 1 to 8 whip-like extensions on them, and they will spin around, which allows the fish to move through the water. And so Euglena and Volvox are things that swim around that are not parasites, but the ones we normally deal with is Ichthyobodo and Spironucleus. On older literature, you might see that called Costia and Hexamida. And so here's what we look like. This is an individual Costia right here. Costia are very, very small. They're the smallest of the protozoa that we normally see, and they have little hairs that stick out there. And their body is shaped like a comma or sort of a dish. It's a saucer that has an indentation in it. So when it swims kind of, like this would swim, and it spirals around as it swims. And so this little organism here, so there's one here, all of these right here are all different costias. And with their flagella, they also have an attachment flagella that they'll attach onto the skin and gills of the fish. And you'll normally find a lot of these on the gills. This next one is Spironucleus. It's also a flagellated protozoa. And on this one, you can kind of see the little shadow here of the whip-like extensions here, the flagella off these. These are sort of cylindrical, and they have a pointed end, and the flagella comes off. And So interesting. Ian, my videos on these aren't
Yeah,
working
I noticed that. Yeah,
because this is actually a video also. And this one,
this one's also a
uh,
video. Looks like maybe
uh,
we're having some technical issues there too.
yeah, so this is the spironucleus in the intestines of an angelfish. This happens to be, and so this darker brown area is the actual intestinal tract, and then all of these little particles that you see here are the protozoa, and they're just swimming around here. They're very robust. The flagella move them along here, and so when you again take a sample of this and look at it under microscope, you're really looking for these microorganisms that are swimming around, and they cause trauma to the tissue of the intestines here, which will cause the fish to have diarrhea or mucus in their stool. So sometimes you'll see very yellowy, thickish, floating stool instead of a dark feces that will drop to the bottom of the aquarium. And so that's due to the protozoa causing intestinal inflammation. So our next group are these spore-forming protozoa, sporozoa, and they don't swim around. They don't have cilia, they don't have flagella, but they live inside of the intestinal tract of the fish, and they do embed into the intestinal lining, and so they cause inflammation. So the fish won't digest their food properly. They usually won't eat well, or even if they're eating, they get thin. Again, the feces get mucousy and yellowish. And if you take a sample of the feces and put it on a microscope slide and you look at it under the microscope, you'll see these little oocysts embedded in there. And they have four sporocytes with two sporozoites. So I'll show you what those look like. And you can treat those with tetracycline in the food. Um, So here's the next group is Myxosporidia, and these are fish parasites that are spread by the things that the fish eat. So annelid worms, such as tubifex worms, which is one of the reasons a lot of people will recommend don't feed eating live food, because the live food can carry parasites that spread to the fish. So if you use frozen food, you don't have to worry about that. And these parasites are very specific to which intermediate host and which primary host they live in, and it requires both of those hosts in order to complete the life cycle. And then these will get into the cartilage and the gill filaments, particularly so for example, the koi fish, some of these like a whirling disease in trout gets into the brain tissue and causes brain damage. We don't see that one in aquarium fish very much, but we do see these. I've seen them in koi and a few other species. So here's the spore we're looking at. This is the fish. It's got the little sporozoites in here. This particular one is Myxobolus koi. And you can see this is a magnified, higher magnification. This is a lower magnification. And this was taken from a gill sample on a koi fish. These aren't very common, but you will occasionally see them when you do diagnostic work on fish. So Microsporidia, the one most people know about is neon tetra disease. It's Plistophora hyphesobrychonis. It's a parasite that gets inside of the muscle tissue or other organs of the fish. But it's not just neon tetras that can get this. It's actually seen commonly in zebradanios in laboratory settings, but it can also go into cichlids or other species of fish. The spores are in the fish. If that fish dies and other fish eat it, that's how they can become infected. So you want to always remove dead fish as soon as you can so that other fish aren't eating them and getting the parasites that those fish have. And they can get this discoloration of the skin, the fins, and the muscles will start getting swollen cysts. If it gets into the spine, it can cause spinal curvature. So these fish usually look just like old, worn out fish. They're thin, they're discolored, they're crooked, and they just don't do well. And unfortunately, there's really no good treatment for this. And as I mentioned at the beginning, a lot of these classifications, these microorganisms are so small that it's really hard to tell for sure what they are. So Microsporidia, which we've frequently or commonly referred to as a protozoa in the past. They're recently thinking, based on DNA analysis, that these Microsporidia may be closer to a fungi or some related other group that's not a fungus or a protozoa, but a new group with a common ancestor. So, we don't know for sure, but it's a parasite or a problem that when it gets on the fish. So those are the main common problems. The saltwater parasites are very similar to the freshwater parasites. They cause similar symptoms, and they're treated with a little similar treatments. So how do you diagnose these? Well, the most important thing is you want to observe your fish, make sure they're active and eating and swimming, and the skin looks good, they don't have any lumps or bumps or discoloration. And then if you start seeing something like that, we want to do a biopsy. And if there's a lot of local veterinarians who work with fish now that can help you with this, or biologists that work in the fish industry. And what we'll do is we take samples from the fins, samples from the mucus on the skin, samples from the gill, and we look at those on a microscope slide under the microscope. That's going to help us find, fungus, bacteria, and other parasites as well, but definitely the protozoa. And each type of parasite may need a different treatment or different treatment protocol because of their life cycle. And a lot of times you'll find more than one parasite on the fish, sometimes lots of parasites, lots of different parasites. So what do we mean by this biopsy? So here's how you take the biopsy. And these are fish that have been anesthetized. These particular examples are koi. Koi are easy to work with because they're pretty big, but you can do this with tropical fish. I do this with guppies and smaller fish all the time. So you anesthetize the fish. Here, this one's out on a wet towel. This one's off on a wet sponge. You don't want it to dry out. You got to keep it wet. Always wear gloves to protect your hand, but it also protects the fish against damage from your hands touching the mucus. So you want to keep the skin wet. Then I just use a cover slip, which is a little piece of glass. You can also use a spatula or even some kind of a blade of some sort, but you're not cutting into the fish. You're actually doing a scraping so the blade would be going backwards rather than forwards. And you're taking a sample of the mucus, trying to get a little bit of the epithelium, but trying not to remove scales. And sometimes on little fish, the scales come off, and that's okay, but you try not to damage the actual scale of the fish. So here's taking a skin scrape. This is a gill biopsy. You can also take biopsies from the fins. You're just using tiny, tiny scissors to take a little bit of the tips of the gills. We're not cutting the gills out. We're just taking samples of the tip. So on this slide, these are the scissors I like to use. These are actually suture removal scissors, and it's kind of hard to see, but if you look at the point, there's a notch in here. That notch is used to get underneath a thread to cut the suture for suture removal, but it works great in grabbing a little bit of gill tissue to take those. So this is a little sample of a gill tissue here. On a bigger fish, you might take more. This is just the tips of some of the gill tissue. On a dead fish, of course, you can take lots of samples, and you would get bigger samples and more of them. And this little slide is to remind you, this is a fish that's floating, it's under anesthesia, and it just happens to be passing a poop. So this is great. Get that fecal sample, put it on the slide, look at it as well. So the tissue is covered with water, a microscope cover slip, this little square here, is placed on top of it, and then it's looked at under the microscope. And here's something you might see. So this is a gill tip biopsy. All of these dark ovals are actually ick parasites. And you say, wow, that's a lot of them. Yeah, they can be very, very plentiful. And you'll see here, this one is embedded inside the gill tissue. These were probably attached to the edge of the gill tissue, and they've kind of come off because of the skin, the biopsy technique, but they'll be embedded inside the skin or here inside this gill tissue. And the other thing about this gill tissue is you see this white layer right here. This outer layer on here is not normal. It should not have that. That's epithelium and mucus that's called hypertrophy. It's building up to try to protect the gill against these parasites. So these parasites are irritating the gills, causing problems. And the fish doesn't like this, so it's developing this protective layer. But over time, this protective layer can build up and actually block the water flow into the gill or across the gill so that the fish can suffocate because of this irritation that's caused by the parasites. And any types of parasites on the gills can cause that. So here's again a little biopsy. This is trichodina here, these little circles. This one is a gas bubble. So one of the things you'll learn is separate air from parasites. The parasites are swimming around. So on this one, what do I mean by swimming around? Well, there he is. He's got this little cilia all around him, and he's just sitting there on the surface of that epithelium. So it's actually this. You can see it's a gill. These little dark spots are epithelial tissue, pigments, cell, and this organism is using its cilia to crawl around or attach to the skin. And then this little denticular ring in here is doing trauma to the skin. And so that trauma then causes skin damage. And then we already mentioned bacteria and fungus can get in there and start growing. So how do we treat these? Well, there's a lot of different medicines that we use for fish, and there's different medicines for bacteria, for fungus, for protozoa, for worms, for crustaceans. And so there isn't really one cure-all that fixes everything. So that's why it's important to make a proper diagnosis. And you may have more than one thing going on, so you may need to use more than one treatment. But usually we'll treat the fish by doing what's called a medicated dip or by a bath. The bath is where you're adding medicine into the aquarium and just leaving it in there. So the fish is constantly bathed in that. A dip is where you have a separate tank or even a plastic bowl. You put the medicine in there, you put the fish in there, you leave it for a certain period of time, 5 minutes, 10 minutes, an hour, whatever the designated time would be. Then you take it back out of the medicine and put it back into the aquarium. Treating the fish is best done in quarantine tanks or hospital tanks that have nothing else in them except maybe an air stone, a heater, a thermometer, and possibly a sponge filter or some kind of filtration. But if you don't have any ornaments or gravel or things like that, it's actually a lot easier to medicate, and you have a known volume of water so you can measure your medication properly. If you don't add enough medicine, it may not work. And if you add too much, it can be harmful to the fish. So it's always important to know an accurate volume of water in the aquarium. A 10-gallon aquarium doesn't hold 10 gallons of water because you're not filling it all the way to the top. You have other things in it that displace water. So you have to know how much water is actually in the tank and then measure your medication properly so you're giving it the right amount of medicine for that volume of water. Some medicines can be added in the food. Metronidazole, tetracycline, other antibiotics are best given in the food. Metronidazole is used for intestinal protozoa, so that's a good thing to use to get medicated food, or you can make your own medicated food. There's a lot of sources on how to do that. And then you have to feed this for a period of time. So a minimum of 3 days, for example, but it might even be longer than that, or you might go 3 days a week, multiple weeks in a row to make sure you're eliminating the parasites. And it's also necessary to repeat the treatments to make sure that all the parasites are gone, and maybe repeat the diagnostic tests to make sure they're gone. So especially we talked about the life cycle of ick, you have to do multiple treatments to get every stage of that life cycle. Whereas some of the other ciliated protozoa or other flagellated protozoa that have simple life stages don't require that long of a treatment period. And so what are we talking about for treatments? Well, the most common one we normally use is formalin. And so if you go to the pet store, the majority of the fish medicines, it's, Riddick, Quick Cure, Curic, whatever they are, they're usually a formalin base, and they'll have formalin malachite green, or formalin and methylene blue, formalin and Victoria green, or formalin and acroflavin, or some other components in it. But for protozoa, that is probably your best treatment at this time. There are some other products that contain hydrogen peroxide. Minfin is a really good one that's hydrogen peroxide and peracetic acid. That works really well as well. For saltwater fish, we typically use a copper compound, so cupramine or other form of copper sulfate. Freshwater, we don't use copper as much because copper is toxic at low hardness and low alkalinity. So in saltwater, we have high hardness, high alkalinity. It's not a big a problem. In freshwater fish, if you're using copper and you have a soft water or low alkalinity water, copper becomes much more toxic. So you have to use it very, very carefully in fresh water. Potassium permanganate can be used, and that's another thing that koi pond people use a lot. So these are all other things that can be used for treating protozoa. And then again, check the fish after treatment to make sure it's cleared, make sure they're healthy, and to be sure that they don't have, bacteria or fungus or other problems, which we will talk about those in a future lesson. And that's what we got for today.
Awesome. Well, thank you, Dr. Nick. There's a lot of great information here, and I think this, like you said, this is a kind of a part of what's to come here in the coming episodes of our Tank Talk. So, thank you, Dr. Nick, for taking the time to go through this presentation. Really, really helpful information. Hopefully, everybody was able to catch back with the live stream. We did have a first part. If you just joined the second part, we had some network issues in the beginning for the first part, so go check that out. We'll work on stitching the two together and putting it back out there. Help you guys out also. But yeah, stay tuned. On the next episode, we'll be continuing discussions about these types of topics. We welcome your input and your feedback, of course. But thank you most of all for joining us. If you have, we will look forward to discussing more Tank Talk topics in the future episodes. Thank you.